Thanks to everyone for joining today's webinar. So just a quick acknowledgement of my colleagues here at the University of Liverpool, including members of the Prenatal Pharmacology group, as well as the funders who support the work that we do. So I put this slide there just to acknowledge the fact that what I will be sharing today in form of mostly proposal, lessons and proposals, they are based entirely on guidance documents and studies that have been published. And I'm sure there are many people in the audience who most likely contributed to this output on which today's webinar is likely based. So I would like to start by asking the question, why is this topic very important? And I think the answer is simple, because the context in which we work has changed. So right now, we're moving away from short-term safety windows to life cycle evidence generation. And you soon see when I share some very recent regulatory documents that are behind this. So right now, the regulatory direction is to move away from default exclusion of pregnant and breastfeeding participants to planned inclusion and or retention if they're already participating and then they get pregnant in the course of a clinical trial. If you think about it, this is particularly important for long-acting therapeutics, which have very extended exposures that are beyond dosing visits and the usual study follow-up period sometimes. So trial windows can be completely outside what the long-acting therapeutic drug exposure windows are sometimes. And sometimes people get pregnant while they are taking these medications. And the image that I put here illustrates why this happens. This is an illustration of what we call the flip-flop kinetics of long-acting therapeutics. So you give an injectable of a long-acting product as an example, or an implant that is long-acting in nature. The release of the drug from the depot controls what you find in the blood. And because the rate of absorption is much more slower than the rate of elimination, then you have prolonged exposure of the drug in the system, like you can see here, compared to conventional therapeutics. So as a result of that, this shift away from short-term safety windows to life cycle evidence generation is particularly relevant for long-acting therapeutics. Developmental susceptibility also change over time. And the windows that need to be investigated sometimes, like the preconception stage, gestation stage, lactation, infancy, and childhood, all of these developmental susceptibility periods, they're completely different and they're not interchangeable. And so the question we should be asking ourselves really is, how do we learn safely across exposure and development of individuals? And just to further illustrate this exposure to outcome map, so what I put here, we have a preconception stage all the way to adolescence. So this is the typical active follow-up window for clinical trials that involve pregnant individuals, whether purposely designed to include pregnant women or individuals get pregnant in the course of participating in a trial. So follow-up generally end around this period, around delivery or early childhood stage, neonatal stage, or very early stages of infancy. However, when you look at long-acting therapeutics, like I mentioned on the previous slide, an individual takes an injection, maybe at the start of pregnancy. For ultra long-acting therapeutics, the exposure to this medication can continue all the way to the neonatal period. And if the injectable was taken during the period of pregnancy, they say the second or third trimester, exposure can continue even all the way to infancy. That is from a single administration. So this is the exposure period. What about outcomes? Outcomes can happen from any time during the period of pregnancy, from first trimester all the way to adolescent and even beyond. So you can see the mismatch there. So what is needed is for us to align this map. so that we have a properly aligned exposure to outcome map. And that should be what informs the study follow-up period. So in today's webinar, I'll take you through a journey starting from a few historical case studies that are very popular, and I'm sure many of the audience are very familiar with. After learning from those case studies, we'll look at how we can apply the lessons from those studies to designing longitudinal studies for safety assessment. We look particularly at retention, biomarker strategy, as well as data integration. And then we'll move on to regulatory expectations and ethical imperatives in view of recent changes in the landscape. And then after that, we talk about real-world evidence and digital tools that can facilitate the strategy that I'll be sharing with you shortly. And then we'll conclude with practical design checklist for long-acting therapeutics, including a very important but hypothetical example, just to illustrate how the strategy that I'll be proposing can work in practice. So let's start with the historical cases. The first one is thalidomide. The case of thalidomide helps us to learn a critical lesson in the fact that short-term tolerability of medication is not the same as developmental safety. A drug is tolerable over a short period of time. We just don't know what might happen. So pregnancy risk cannot be an afterthought. That is a key lesson from the story of thalidomide. It's a defining event in modern reproductive safety regulation because most of the regulations that we have right now, they result from the tragedy of thalidomide. But I'm not going to dwell on the details, but on the lessons. And part of the lesson from thalidomide is that preclinical and clinical systems that were in place at the time, they failed to anticipate pregnancy-specific harm from that medication. Timing of exposure and endpoint selection were very critical. So if we miss the critical timing of exposure and we don't select the right endpoint for our study, then we would miss potential outcomes that would be very relevant. And over the last so many years, a lot of people have done studies. So right now, we have a fairly good movement towards understanding the mechanisms behind what was observed with thalidomide. So for long-acting therapeutics, there is no way for us to discontinue if a pregnant woman takes a long-acting injectable or implant as an example. There is no way in most cases to quickly retrieve it. And so exposure can continue even until the baby is born. And that makes it really critical for us to prospectively plan evidence generation system so that we can capture all the relevant outcomes. So that's the first example that I want to share. The second example is Diethylstilbestrol, or DES, about delayed outcomes and long latency that often occur in some cases. And the lesson here is that the absence of early harm is not proof of developmental safety. A medication is given, baby is born, baby looks fine, happy, drug is good. That is not always the end of the story. For DES, prenatal exposure produced outcomes that were recognized many years later. In this particular study published a couple of years ago, it analyzed data from so many different studies about this medication and the age-specific incidence rate for cervical and vaginal cancers. It was found that in most cases, the vast majority of cases came to develop around the age of 20 years of age after in utero exposure to DES. But interestingly, the same study also found that there was actually a second spike in exposure in the outcome around the age of 40. So 20 years later, and even 40 years later, there were still cases that were associated with in utero exposure to this particular medication. So the key lesson is that birth and infancy are not always sufficient observation windows for assessing safety of a medication that has been taken during pregnancy. And so a clean early signal does not rule out that later reproductive or oncology outcomes may become known. So follow-up duration should be about the biology and not about a convenient study design. And that takes me to the third example that I want to share. And this is regarding antenatal corticosteroid. The lesson here is that short-term benefit and long-term outcome must both be designed for. In this analysis, this is a systematic review of so many different studies connected with this particular example. Extremely preterm children were found to benefit, so they actually have some protection from neurodevelopmental impairments. But when you compare that to full-time children, you find that there was an increased risk of mental and behavioral disorders, as well as increased risk of neurocognitive disorders. So you see, very interesting mismatch because of the gestation context. And the lesson include the fact that short-term neonatal benefit cannot be the end or should not be the end of safety evaluation. Long-term child outcomes should be pre-specified. It's not always the best when it's an optional extension to an ongoing study. It is best when it is planned for. And gestation context, dose of the medication, the time of exposure, and the comparator are all very important in study design. So we do not let neonatal endpoint close the child safety file. We should leave it open for much more longer. And I'll provide some potential strategies of achieving that as we go along. The next example is more recent. This is the story of Dolutegrvir in the Tsepamo study in Botswana. For this particular study, the key lesson, or this particular case, the key lesson is that early signals need refinement, not panic or paralysis. And so it becomes important for us to have safety systems that can detect signals, but not just that, but systems that are also capable of stabilizing the signals as quickly as possible. In this particular situation, Dolutegrvir was associated with early neural tube defect. That was the early signal, and it had a major impact on policy and access for people who really needed this medication during pregnancy. This is a summary from this particular study published recently that looked at all the different cases and how the numbers have changed over time. So we're moving from a really high signal, the green bar line that you see there, that is cases from individuals who were exposed to Dolutegrvir during pregnancy. And these are other antiretroviral drugs. So you see the massive difference between the two categories. But you notice that as the numbers of individuals exposed to Dolutegrvir during pregnancy increased, then you see the signals dropping. So the signal dropped all the way to the same level as with other antiretroviral therapy, because eventually the system was able to stabilize the signal. So the key lesson there include the need for rapid denominator strengthening and transparent uncertainty communication so that there is no panic that could potentially affect policy and access, as well as proportionate decision making. So all of these are lessons that we learned from the case of Dolutegrvir in the Tsepamo study. So this is a summary of failure modes across so many different studies, not just the ones that I've shared on these slides, but also from so many other cases that many of you might be familiar with. Misspecified exposure windows, follow-up period in a clinical trial that ends before outcomes became visible, weak denominators or inconsistent outcome definitions, a lack of integrated maternal-child data infrastructure, and lastly, poorly prepared communication when signals evolve. So strategies that we need for the long haul will need to be such that they're able to overcome as many of these failure modes as possible. So let's now move on to the second part of my presentation, and that is, how can we apply these lessons? How can we design longitudinal safety studies that are not associated with the limitations I highlighted previously? So the key point here is not to start with a convenient sample. And what I mean by that is we do not want to build a cohort that is easy and available and merely operationally convenient. That is not the solution. So the questions we should be asking is, what exposure window is biologically relevant? Which mechanism is hypothesized in terms of drug exposure? Is there fetal exposure? Is there placental exposure? Could there be an impact on the placenta? Does the drug cross the placenta? Is it transferred into breast milk? Does it affect the immune system? All of those questions. And this impact, any potential impact, any mechanism that may be associated with potential impact, is it organ specific? And also, which outcomes should appear early or are likely to appear early? And which ones are likely to appear much more later? And so we require some kind of staged follow-up. And which comparator best addresses the indication that we're interested in? What about disease severity and co-medication? So it's really important to think about all of this while designing a study. So the COSA question should come first. So the question we should be asking is, which outcome window, which population, which comparator, which outcome structure are needed to answer the biologically meaningful safety question that we're asking? The question we shouldn't be asking is, who can we recruit most easily? That is the wrong question to ask. So starting with the COSA question is really an important part of the strategy. So the second component is the quad architecture itself. It needs to be built for the long haul. And this is where it becomes really important to think in layers and not one monolithic follow-up study. And just to explain what this means, we need to design follow-up as a portfolio. Broad core outcomes for all the participants are collected, and then we collect deeper phenotyping variables in focused subcohorts. And this image here illustrates what I have in mind about this. The first layer is the maternal index cohort, and that really is the core cohort that a study might start with. But on top of that, you then have a second layer, and that is the pregnancy or birth model of the study. And this, why the first one helps you to assess the impact of the medication on the indication, on the disease severity, and so on. You can assess PK, you can assess the impact of the drug in obstetric or context, and so on. The second layer helps you to look at fetal growth, any impact on delivery, and so on. And then the third layer is the infant and child model. And this is where you can follow up for growth, neurodevelopment, and other outcomes. But there is a fourth component to it, and this is a passive linkage layer, which can cross all these other three layers and do a lot more than it. And this includes things like electronic health record and registries and similar reward evidence or data sources. So all of these constitute the third layer in this quad architecture for the long haul. So as we go along, you will see in the example that I'll be presenting how this might work in practice. So, but the key point here is that this type of design needs to support operational feasibility and scientific depth by separating core follow-up from nested mechanistic sub-studies. So the next component is the need to treat retention as a scientific variable. And that means it needs to be designed for and not assumed. And the reason for this is because loss of follow-up tends to be a really major issue in a clinical trial. And it's often non-random and can be socially patterned as well. And so it becomes really important to take care of that. It can affect the interpretability of a trial data. And so it's not just about sample size. It really should be treated as a scientific variable. And follow-up burden should be prioritized. Call visits are very important, and so that's a priority area for follow-up. Remote contact are very important. Local care and targeted deep phenotyping another areas where follow-up burden might need to be prioritized. Modern quality by design thinking that you find in modern regulatory documents support treating retention as critical to quality factor. So it's really very important to look at follow-up beyond trying to get the right sample size that we're interested in. And there is another important point that consent and recontact strategies must anticipate the fact that some of the participants will get pregnant and babies will become infants and an infant will become a child. And so it's important to really make sure that the strategies will develop for consent and for recontacting the participant needs to take all of that into consideration. And then we design a protocol that will suit what we've anticipated. So the next point I want to touch on is the biomarkers strategy. It needs to be such that it's aged with the child. And there are a number of components here, but the key point is that our biomarker strategy needs to triangulate exposure, mechanism, and outcome. It shouldn't be just like a shopping list. A biomarker should be selected because it matters for the exposure we're interested in, and the exposure is linked to a particular mechanism that is linked to a particular outcome. And the exposure, when it comes to the drug, it could be any of this. Or the mechanism could involve the placenta, it could involve the immune system, it could be endocrine, inflammatory, it could be organ specific, and so on. And the outcomes could be growth, neonatal adaptation, neurodevelopment, organ function, and response, and so on. And biomarkers need to be interpreted by developmental stage. It shouldn't be interpreted as a one-size-fits-all child safety measure. Remember what we discussed regarding the case of antenatal corticosteroid. So that's a classical example that teaches us that our biomarker strategy needs to be developmental stage specific. And just one last point regarding long-acting products, the sampling time points may be as important as the assay itself because of the long exposure period that a child might be exposed to in utero that might even continue during the breastfeeding period. And the last component in longitudinal study design that I want to touch on is data integration and analysis plan. So it's important to reframe the study as an evidence system and not just a study that is isolated from what comes before and what comes after. So we need to define a minimum common data set and data dictionary even before the first participant is enrolled. And where governance systems permit, it's really important to link the clinical trials with registry, electronic health record, and pharmacovigilance systems. And you see the reason why this is really important. And the only way to link is really to establish a system where, whenever possible, there will be an identifier that will be replicated across all of these systems to make it easy to link the records. And so it's important to pre-specify methods for handling or co-funding, because these tend to be a major issue with reward data sources. But if we specify methods for handling them, it might be a lot easier to account for them. And whenever possible, we use model-informed approaches, including POPPK, PBPK modeling, pediatric extrapolation, exposure response. So all of these model-informed approaches can really generate really valuable data that will be useful for designing aspect of the study that will point us in direction of where follow-up might be needed to be more intensive. So that is about how to apply those lessons in designing long-term longitudinal studies. So the next part is regarding regulatory expectations and ethics. Now, the current guidance regarding clinical research when it comes to pregnancy and lactation and children has really moved on. And these are examples of some of the guidelines that have been released in recent years. And one thing that is common to all of these documents is they point in the same direction. And that is the fact that evidence generation needs to be planned. It needs to be proportionate. It needs to be scientifically justified and also capable of supporting decisions over the product's life cycle. Like I mentioned at the start, we've moved away from the era where pregnant individuals are excluded from clinical trials. But now we need to start planning for inclusion. Then when it comes to non-clinical and translational readiness, what reviewers would expect you to have thought through include some of the points that I put on this slide. And the first has to do with DAT studies, because this remains central to pregnancy-related risk assessment. And they are still the basis for making a decision on whether pregnant individuals should be allowed to be enrolled or whether they can be allowed to continue in a clinical trial. And so it's important to assess exposure duration, the reversibility of exposure, placenta transfer of a drug, or milk transfer, as well as developmentally sensitive organ systems. So these are things that should be assessed as part of that studies, which tend to use one non-rodent and one rodent as a species. But more recently, PBPK modeling, new approach methodologies, these are more human relevance in vitro methods like organoids and organ-on-chip systems. These can really fill important gaps in the translational relevance of the data from that study. So they can replace a lot of endpoints that studies can generate. So this needs to justify the timing, the sampling, and the follow-up intensity that are put into clinical trial protocols that anticipate enrollment or retention of pregnant individuals. For instance, you can have a new approach methodology assay that assess the impact of a particular drug, of the drug you're interested in, on a particular specific organ that is developing. And so if data from the NAM study indicate that the drug has no impact on a specific organ that is developing in the fetus, that could go into the justification for allowing pregnant individuals to continue. And it could be the reverse as well. And same thing with PBPK models can generate data that relates to drug exposure, maybe indicating that this drug does not cross the placenta. There is extremely minimum chance that the fetus will be exposed to it. So all of these can generate evidence that can inform the timing, the sampling, as well as the follow-up intensity of the clinical trials that follow. Then the ethics imperative. So routine exclusion, what it does is to move uncertainty into clinical care rather than eliminate the risk. It never takes care of the risk. It only amplifies it within the clinical care context. And so risk proportionate inclusion requires clear benefit risk logic and staged communication when any signal is detected. And so maternal, fetal, and infant interest should be considered together, not as competing abstractions, because they are all important. So long-term child data stewardship, privacy, and management of incidental findings should be explicitly put into the trial protocol. These are some questions that reviewers should be interested in. And these are cross-cutting questions that clinicians, trialists, regulators, and drug development teams should look into. For instance, why is this follow-up duration? Why is it biologically and clinically justified? Which outcomes are critical to quality in this particular clinical trial? And how will they be measured? What non-clinical PK and exposure data support inclusion or retention of pregnant individuals in the trial? So they look at the non-clinical data, they look at the PK data, look at exposure data, whether from DAT studies or from new approach methodologies and in silico methods. They look at all of those. And the last question is, what triggers enhanced follow-up? Risk management plan updates, or post-authorization safety studies, or signal escalation. So they need to pre-specify things that will trigger any of these measures. And if there are uncertainties, maybe because of a signal that has not yet been stabilized, like seen in the case of Dolutegrvir, how will it be communicated? So that needs to be planned for. So the next part, which is the final part of the presentation of the webinar, is how can we make this happen? So real-world evidence needs to be looked at as a planned infrastructure because it's a critical aspect of implementing a longer strategy. Because we need to recognize that no single study can do all of the work. So it's important to then use real-world evidence as part of the initial design of the study, not as a rescue strategy after the trial has ended and only during the post-marketing period. So it's important to plan maternal-child linkage across the different data sources that will tie them, whenever possible, to the clinical trial data. Then it's also important to use distributed networks where single study sample size is often insufficient. Then it's important to document provenance, data quality, and denominator logic. So all of these are things that would help to strengthen any real-world evidence that needs to go into the longer strategy that we're trying to discuss. Then the next is digital and decentralized follow-up. So this can facilitate sustainable follow-up without diluting the scientific value of studies. And these are some examples which can reduce participant burden, and they really need to go into this type of strategy. So digital tools, for instance, they can support remote questionnaires, uh, caregiver diaries, wearables, biosampling logistics. So digital tools can help to facilitate all of this. And hence they can make things that would otherwise be impossible, they can make them a little bit more manageable. But fit for purpose validation is essential for digital endpoint. And it's important to avoid digital exclusion. And the only way to do this is to maybe think of how to provide low-tech solutions, language supports, and local alternatives that trial participants will be um, familiar with, or they will find it easier to use. So these are some of the things to consider when it comes to digital health or technologies and decentralized elements um, of follow-up. They need to be fit for purpose, participant-centered, and governed with the same standards as site-based data capture. So how can this then be implemented in practice? So the key ideas include combining one core protocol follow-up with extension follow-up, passive plus triggered surveillance, and signal response plan. So the design principle include the fact that it is feasible for us to have a long-haul system that combines all of these different components. Intensity of follow-up can reduce with time, but the chain of interpretation must remain intact. And that's really the key point. And that's why it's really important to establish a strategy to link the different data capture systems together. So you can just to illustrate this, so you have your core protocol with a lot of governance involved in it, a clinical trial where you have pregnant individuals, you're able to follow them to delivery, maybe early infancy as well. But then beyond that, it's important to establish a hybrid extension, which could include remote and local follow-up for up to 5 years. This may require reconsenting, recontacting. The next stage will be a passive linkage system. And this could be strategies that would combine different reward data sources like electronic health records, registries, surveillance systems, and so on. A critical aspect to make this really useful is common data model. So this is very important to make sure that evidence that is generated across the system can be combined and used to inform any analysis that one may plan to have. So for passive linkage system, most long-term follow-up can happen passively through linked routine data sources, but pre-specified safety signals or event trigger more active targeted follow-up. So what I mean there is most of the follow-up, they happen passively over time. But when safety signals that are already pre-specified in the study design, when those events happen, then they trigger more active and targeted follow-up. And then we have a signal response. So it's important to have a signal response system and make a decision on when escalation will be necessary. So this really involves a very good participant communication strategy. So just a few questions that may be on your mind. And the first one is, if we're to align study design with the strategy described here, would the alignment require a pregnancy-specific trial? Does that mean we need to have a pregnancy-specific trial so that we can have a longer strategy to monitor? So the short answer is not necessarily. Full alignment does not mean that we have that type of arrangement, but it does require pre-specifying and adequately resourcing and analytically credible pregnancy to childhood evidence pathway. And I will illustrate that shortly. It might mean that we need to have a tiered approach, and I think that will be more practical. And then we align that with current regulatory thinking. So just to illustrate the fact that a tiered approach actually aligns with present regulatory thinking, for many long-acting products, for instance, tier 1 might be sufficient. And that has been the recommendation in many of the guidelines. And that is a situation where you retain participants who become pregnant in the course of a trial. So that is tier 1. And so it's important to pre-specify exposure timing, obstetric outcomes that you're interested in, neonatal outcomes, breastfeeding capture, as well as reconsenting as needed. The second tier might be necessary because tier 1 may be insufficient when the use of a particular long-acting product in pregnancy is likely. So when it's going to happen, and there is no doubt that it will, and exposure is difficult to reverse, like is the case with long-acting injectables and most implants, and fetal or infant exposure is plausible, or PK dosing uncertainties remain, then tier 1 is not really the strategy we should be looking at. We should be looking at tier 2. And this is embedded pregnancy and lactation sub-study within the core cohort. And this will require these particular biomarkers for assessing exposure. Or tier 3 may be important in some cases, and this is a prospective pregnancy cohort or registry study which will provide access to larger denominators as well as rare outcomes. And last is tier 4. This is for pregnancy-specific indications. So this will be needed if it's for a pregnancy-specific indication. So intentional use is what is important in this particular case. So you can see clearly there that it does not necessarily require having a pregnancy-focused clinical trial. So any of these tiers will provide us with opportunities to have a longer strategy. And then the last question is, would this delay approval or time to market for the long-acting product? Well, the strategy should actually de-risk approval. It shouldn't delay access until late childhood when all the possible outcomes are now known. So the key is really to have a balanced model. And the balanced model will be one that enables approval, generation of evidence that would enable approval, followed by pregnancy-specific pregnancy or lactation pathway, and then post-approval real-world evidence and child follow-up after that. There are potential benefits of this strategy rather than disadvantages. So there are fewer late regulatory surprises when it comes to pregnancy and children. It provides opportunity to have a more focused drug label and regulatory guidance regarding the use of the medication in this population. It also enables faster time to implementation because it's not just about approval, it's also about implementation in the right population. And lastly, it enables generation of parallel evidence through registry and all the other data sources that I mentioned previously. There are some trade-offs that we need to be that we need to have in mind, including possibility that the upfront cost might be high, but that is only possible if we make it too complex. Slow study startup, but that is only if the design is overly built. There is also a risk of unrealistic pre-approval expectations and large post-approval commitments if all of these other data generation plans are overbuilt. So here is a hypothetical example of how this might work in practice. This example relates to a once yearly ultra long acting medication. And that's the product premise. So you start with pre-IND, non-clinical studies, which will include DAT studies, as well as NAMS, in vitro models, and maybe some PBPK modeling. And you use all of these to generate evidence that you use to support staged or planned inclusion of individuals who may get pregnant during the clinical trials. And then you need to pre-specify triggers for more intensive follow-up. It could be when somebody gets pregnant, if the level of infant or fetal exposure is predicted or seen to be much more higher than you expected, or there is a cluster of sickness detected somewhere. So those need to be pre-specified. And then phase one. So phase one, the usual phase one clinical trials, that's when core clinical trial visits will start. That will provide opportunity for generating data on tail phase PK of the medication, to plan what happens if someone missed a dose and so on. And then phase two will provide opportunity for embedded PK studies or lactation sub-study if you were going to tier two. And then at that stage, hybrid extension can start. And that's when follow-up begins to become less intensive. And of course, you've got your phase three trials, which are pivotal for getting approval. So approval of the study can be obtained, but all of this follow-up will see continue, especially through the passive linkage ecosystem that you've already established. And then drug gets approval, but life cycle evidence generation still continues, as previously described, through all of these systems that have already been built. So as you can see, the real idea is that follow-up intensity can reduce over time, but the safety system does not disappear. It is retained. So that's how I think this strategy can be implemented in practice. And so these are the five take-home messages that I've got for you. The first one is the fact that long-acting exposure requires long-term evidence plans. I think that's very clear from what we all understand. And the reason is simple. So someone takes a medication at a certain point during pregnancy, exposure does not necessarily end a couple of days later or even a couple of months later. In many cases, it continues even until the breastfeeding period. And the second is what we learn from history, that missed delay harm can happen. And signals can be override if we look into them before the denominators stabilize. And the third one is that timing, retention, and data linkage are scientific design choices, and they should not be taken for granted. They should be well planned. And we've got resources, we've got tools in place to do some aspect of this, including model-informed approaches. And number four, regulatory expectations have shifted. And now there is a favor movement away from exclusion to planned inclusion of pregnant and lactating individuals. But that needs to be coupled with proportionate life cycle evidence generation. And lastly, a practical solution for this strategy is an integrated evidence system. Our trial follow-up, registries, real-world evidence, biomarker strategy, and signal governance need to be designed from the start to really enable us to generate evidence that we need over a long period of time. So designing for the long haul would help us to safeguard maternal and child health while at the same time enabling innovation. These are a list of things not to do. We should not define a single undifferentiated childhood safety window because there are so many different stages of child development. And the same thing with the gestation context. And we do not want to rely on one registry as the entire long-term plan. And please don't collect rich neonatal data, but then fail to collect preschool outcomes when later development is relevant for the safety questions that you have in mind. And it's important to not use digital tools like a showcase that, yes, we've got a really nice tool, so let's use it. We should assess accessibility and have a validation plan in place so that the generated data will be as useful as those that are collected in connection with in-person follow-up. And of course, we do not want to wait for a signal before planning how to communicate what the signal means and what it does not mean to the population that need medication. So that way, we protect the population and we speed up innovation with long-acting therapeutics. So thank you so much for your time.